The rate of enzyme reaction. So in the lab today, we're going to kind of break it down into two parts for you guys. So we've been talking about enzymes, and we know that enzymes are a form of a catalyst, and there are other things besides proteins that can be catalysts as well. But enzymes are a special type of protein that are going to speed up reactions for us. We've already done an activity where you guys were the enzyme, or you saw me be the enzyme in the video, and we know that an enzyme is going to work upon a substrate in order to make its product. And we know that each enzyme is specific for the substrate that it works upon, and it can work in both directions. In other words, in the video I kept saying, oh, the toothpick represents maltose, a disaccharide, and when that enzyme acts upon it, maltase being the enzyme, what we're going to see is that our product is glucose and glucose. Well, if we had a way of putting that two toothpick pieces together, we could say, oh, glucose and glucose makes maltose. It's the same enzyme that's doing that, still maltase. That's going to go ahead and utilize that. So today what we're looking at is within this enzyme substrate complex, we want to look at the effects of temperature as well as pH and the roles that they play on the reaction rate of the reaction that we're about to see. So we know that temperature will affect enzymes, and we know that body temperature is the most specific, or probably the best temperature for these enzymes to act in. So usually around 37 degrees Celsius, 98.6 degrees Fahrenheit, these reactions should run very, very quickly. When we start to get a little too high or we get a little too low in terms of body temperature, it's going to start to slow down our enzyme reactivity, and eventually, because remember, these are proteins, and this enzyme is a tertiary structure, so we talked about our primary structure, strand of amino acids put together, our secondary structure where we have beta and alpha helixes, and then our tertiary structure, we have these disulfide bonds between these layers. What happens is that this tertiary structure begins to break down, and then the reaction does not occur very well. Well, if it gets really high or really low outside of that optimal or best area, they do what is known as denature, where they kind of completely unravel and they don't work whatsoever. And a lot of times they'll be destroyed in the process. So what does that mean? Oh, I don't have that protein in me. I don't have that enzyme anymore. We have to go back to my DNA. We have to find the code that makes that. We have to go through protein synthesis, and we'll see that in our next section, and make more of that particular protein if I have those amino acids within my body. It goes back to eating a balanced diet. So what we're looking at today, the reaction. So think about this. We are going to be utilizing hydrogen peroxide, H2O2. Hydrogen peroxide is made within our bodies during cellular respiration. It's actually very toxic for our cells, and our cells need to break it down in order for us to survive. So we actually have an enzyme within our body, as do other organisms, that take that hydrogen peroxide, H2O2, and that enzyme is going to break it down, or the enzyme is going to help speed up the reaction to break it down into water as well as oxygen. So two substances that are no longer toxic for our body. And the enzyme that's going to help to do this is an enzyme known as catalase. You guys can see it right here, ends in ASE. So this catalase breaks down hydrogen peroxide, helps to speed up the breakdown of hydrogen peroxide, which is toxic, into two smaller molecules that are no longer toxic to us. And what we're going to be using today, when we're looking at temperature as well as pH, and we'll talk about pH here in a moment, is we're actually going to use yeast. So on that second page, you guys will see that. Yeast is actually a living organism. So you can buy it from the store. It comes in a container like this. It's very granular. So looks like that. It's a living organism. It's dried at the moment. I put it with a little bit of warm water, and I gave it a little bit of sugar because it needs some food. And I kind of made it a little happy home. He's not very happy right now, but we'll see what happens. And he looks like this. So this is what we use in order for us to make things like alcohol or bread. We allow it to ferment, but it is a living organism. So as it's sitting there, what we have is we have an enzyme catalase in it. So that enzyme catalase can be found in our living organism of yeast, and that's what we're going to end up utilizing today. So we're going to fill these little tubes with different hydrogen peroxide solutions, and we're going to fill them to the top. And when we're looking at pH, we're going to look at pHs of 3, 5, 7, 9, and 11. And within these particular solutions, and you guys will see them on my table here in front of me, and within these particular solutions, we have hydrogen peroxide as well as specific pHs. So remember, 3 is very acidic, 5 is slightly acidic, 7 is neutral, 9 is slightly basic, 11 is extremely basic. So besides just the pHs, I also have hydrogen peroxide in them, so that when I take that yeast and I introduce it to it, what we should see is that the catalase within the yeast should break down the hydrogen peroxide, as long as that protein is not denatured or that enzyme is not denatured, and it should actually have the reaction go very quickly. So we're going to time some things here. So how do we know what's going on, or how do we know that my yeast is physically doing its job? When I fill this little tube with my particular solutions that have hydrogen peroxide in them and their little pHs, I'm going to put yeast within it, but I'm going to do it using this little piece of filter paper. So I have these little discs here of filter paper, and we want to make sure we only grab one at a time, otherwise it's going to change the mass of what's going in there. And what I'm going to do with this particular filter paper is I'm going to saturate it in the yeast solution. I'm going to dip it on both sides and get the excess out. So now what I have is I have this little bit of yeast with catalase here. I'm going to be dropping it into my tubes that have water. Let's just give you guys a little example here. I'm going to put just some of my solution of one of them in here. I'm going to fill it up to the top. There's my little tube of water. And I'm going to take my little yeast and I'm going to drop it in. And what's going to happen is that my little yeast is going to sink to the bottom in the little filter paper disc. And when it does, we're going to start my timer. And what we're looking for is for that catalase within the yeast that we just put on that filter paper disc to break down hydrogen peroxide. So remember, when it breaks down hydrogen peroxide, it's going to break it down into water. Well, we won't really know because my solution looks like water. But it's also going to produce oxygen, and that's the part that we're going to be able to measure. As it produces oxygen bubbles, those bubbles basically trap onto that little filter paper disc and it will raise it to the top. So the faster my reaction of that catalase breaking down hydrogen peroxide into water and oxygen, the faster that disc should rise to the top. So we're going to start my timer when the disc hits the bottom, and we'll stop my timer when the disc comes to the top. For each one of my solutions, 3, 5, 7, 9, and 11 in terms of my pH. Anything over 4 minutes, we're just going to call it at 4 minutes so we don't sit here all afternoon. We're going to do 3 trials for each of them, and then you guys are going to be averaging them out. So I'm going to do 3 trials for pH of 3, 3 trials for pH of 5, 3 trials. I'm going to try to do them all at the same time for you. So you guys will see that you have a little data chart on your particular lab. And I actually asked you guys to create your own data chart, but mine's on 2 separate pages. Yours is all in the one Google Doc on the computer. So my first problem, because this first part we're doing our pH. My problem is listed there for you. It says at what pH will the optimum or the best, so at what pH will the optimum for the action of the enzyme catalase in yeast to degrade the harmful substance of hydrogen peroxide into the products of water and oxygen. In other words, at what pH will my enzyme action work the best, and why? Now don't just tell me about one of them, tell me which ones are going to completely work, which ones won't work at all, which ones will work best, which ones may sort of work, and whatever else you guys have. So what will be the best pH, and what will occur at the other pHs, and why? Remember, no I think, no I believe. If you have yet to make a hypothesis for your pH, stop the video now. Make your hypothesis, then hit play again. So where we're going is I'm going to kind of shoot on over here to my little setup where you guys are going to see my trials now. Let's kind of scrunch you out a little bit. So I'm going to have my 3 trials. I'm going to run 1, start it, 2, start it, 3, start it. They each have their own little timer. Got my little waste bin here. And I'm going to start with pH of 3. Okay, so we are doing our pH right now. I want to make sure everything is out of that little vial that I have. So we're cleaning it out, putting it in my waste container. And there are my 3 vials right there. So I'm going to take my solution of pH of 3. So it's got some acid in there. It's also got some hydrogen peroxide that the catalase should be able to work upon from my yeast. And I'm going to fill those 3 tubes to the top as close as I can with a pH of 3. So you're going to notice each one has their own little stopwatch down below. That way I can run all 3 trials at the same time. I'll start 1, then 2, then 3. And hopefully they're all close to each other in time, but that's why we are averaging them. So what could affect my times? Well, maybe I have 2 pieces of filter paper stuck together. Maybe I've got a little bit more yeast on one than the other. Maybe there's a little bit more solution in one than the other. So there's a lot of factors that could take place within this. So I have my yeast, I have my little solutions. I'm going to take my little filter paper, one piece at a time. I'm going to dip it into my little yeast solution, get the excess off, and I am going to drop it into trial number 1 tube. I'm going to let that sink all the way down to the bottom. When it hits the bottom, I'm starting my time. So we're going to let that time run. I want to do the same thing for trial number 2. My little yeast on my filter paper disc. As much off as I can. Same thing. I'm dropping it into the hydrogen peroxide with a pH of 3. I'm starting my time. My third one, same thing. So all of these are a pH of 3. It's just with us doing 3 trials. We want to make sure that we are running all 3 at the same time, so this video is not super duper long. If you guys were in class, you guys would be running 1 trial, and you guys would then be sharing data with other groups. So we're going to kind of let that go. Right now, this is at a pH of 3. My little discs are at the bottom, and we are going to let them sit there for a little bit. I'm going to get some warm water from my yeast here, so that it can be happy as it's sitting there. The colder it gets, the less it's going to want to grow and ferment. So we're going to make sure it's nice and warm throughout this process. So remember, 4 minutes is going to be my maximum time that we have here. If nothing happens within those 4 minutes, we're going to go ahead and just say 4 minutes. So while we're waiting, my little yeast is going to be going in some warm solution. And we're waiting for those discs to float to the top. Now remember, the discs would be floating to the top because my hydrogen peroxide, which we know is in each one of those tubes with a pH of 3, would be broken down into water and oxygen. And what would be happening, that enzyme, as it's breaking it down, that catalase, as it's breaking down hydrogen peroxide, those little oxygen bubbles would be trapped on that disc and would be forcing it up to the top. So it's going to float up to the top, hopefully. If it is not working, maybe my enzyme's not working. Well, why is my enzyme not working? Well, we are in a pH of 3 right now. So my first one, we're up to about 2 minutes. 4 minutes is my maximum. 1.48, 1.31 is what I have. All of these are my pH of 3. And we'll keep letting them go. We're keeping an eye on them. We want to see if they kind of float up to the top. Once again, my little yeasty guys, they are in warm water. We're trying to keep them warm and trying to keep them happy so that we can let them grow and so that this reaction can work. Trying to give them a little sugar. And we'll let those timers go. Keeping an eye on them to make sure that those little discs not make their way to the top. So we're almost at 3 minutes on that first one that you guys see. We're letting it go. So my enzyme catalase, my substrate would be hydrogen peroxide. My products would be oxygen and water. And oxygen's the only one that we're really going to be able to tell because that would make little bubbles for us on our filter paper disc that contain my living organism of yeast. And that yeast is making catalase, just like what we would, but I don't want to stick us into those little vials and see what happens. So we have our yeast in there with the catalase in there. And we're at 340 right now. We still have no action on our pH of 3, 325, and 306. So we're going to be calling this here in a moment. 350, we're going to say our maximum time is 4 minutes. So we're going to call the first one 4 minutes. So trial 1 for pH of 3, 4 minutes. I'm going to dump it out in my waste container so that I'm good to go with my next round. We're almost at 4 minutes for the other two. We're at 4 minutes, so trial 2 for pH of 3 is also 4 minutes. And a few more seconds to go here just to make sure we have no movement on that filter paper. It's still sitting at the bottom. We're at 4 minutes. So I'm going to stop it. So what you have for a pH of 3 all the way through trials 1, 2, and 3, we are at 4 minutes for all of them. So I'm resetting my timers. I've got my tubes here, timers are here. We are moving on to a pH of 5. So now we are on a pH of 5. It is slightly acidic. I'm going to fill my little glass cuvette tubes up to the top. So that is a constant, same size tube, same type of solution, same amount of solution. My filter paper discs are constant. The only thing that's a variable within this particular experiment is the pH solution that it is in. So whether we are 3, 5, 7, 9, or 11, that would be my variable that you guys have there. So now I have a pH of 5 in each one of my tubes. I'm going to go back to my filter paper discs and do the same thing that we did before. And we know we were a maximum of 4 minutes. So hopefully we get a little action going on here. Stick it in my yeast solution. I'm going to drop it in. We're going to let it hit bottom before I start that time. So it's going down and we start. I'll do my second one for you guys, and we're keeping our eye on that to see if it makes its way back up to the top and how quick this reaction occurs if we have any reaction right here. Same thing, we'll let it sink down to the bottom and we'll start our time. And for trial number 3 for a pH of 5, same thing. Drop it in. And hit our timer. So same thing. Now we are at a pH of 5. It's just slightly acidic. Will this have any effect on our catalase here, or our enzyme catalase that we have within the yeast that's on our filter paper disc? So we're looking to see if we have any type of bubbles that are forming. Let's see if I can get you guys a little closer here. So there's my little reaction going on. So there's a little bit of bubbling going on there with all of them with this pH of 5. But we want to get enough bubbles that it will trap on that particular filter paper disc and it will actually start moving upward. And when it starts moving, or if it does move, I'll let you guys know. So we know for pH of 3, we were at 4 minutes all the way across. And here comes our first disc. If you guys are watching that, our first disc is kind of making its way up. It's floating in the middle. We're gonna let it go a little bit longer. He's kind of just suspended in air there. Not quite what we need yet. We need to make its way up to the top. And it's actually making its way up to the top now, which is what you guys are seeing. And it's there. So I'm gonna stop time. Trial number 1, 1.51. So 1 minute, 51 seconds was trial number 1 for a pH of 5. But we have 3 trials running. So we want to see what happens with trial 2 and trial 3, and we're waiting for that to go up. And we're waiting for those discs to move. So you guys should be writing this information down. And we're still kind of waiting for those 2 other ones to go. Here comes the second one, kind of bobbing around in there a little bit. Third one's bobbing around a little bit in there as well. And we want them to make their way all the way up to the top. Here comes the third one. And my third one's there. So 2.14 for trial number 3. Trial number 2 is kind of taking its time there. We'll let it go. Trial number 3 was 2 minutes and 14 seconds. Trial 2 is definitely trying to work its way up there for pH. It's kind of bobbing around. We have a maximum of 4 minutes. We're at about 250 right now. And that's why we're doing the average of 3 trials. So So still waiting for it. It's kind of coming up, working its way there. Here it comes. Working, 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 and time. So now we're at 3 minutes and 17 seconds for trial number 2. So a little bit of a wide range there. That's why we want to average them. So I'm going to dump out my pH 5 solutions. Now I'm going to go to a pH of 7. I want to reset my timers. So once again, trial 1 was 1.51. Trial 2 was 317, and trial 3 was 214 for a pH of 5. So now where we are is now we are on a pH of 7. pH of 7 is my neutral solution. Now remember, we have different pHs all throughout our body. So enzymes within our body, depending upon where they're located, are going to work in different pHs for us. So this particular pH of 7, let's see what we have here. So we have a hydrogen peroxide solution that is neutral in terms of our pH. And I'm filling these up all the way to the top. So we have those 3 full. Now we're on that pH. I'm going to reset my timers. I'm going to do the same thing. Same yeast, same discs, same timers, same everything. So I'm going to grab my first filter paper disc. And it's real important to make sure that we only have one, because like I said, if we have 2 of them and they're kind of stuck together, then it's going to change our results. We're dropping our first one in. Let that go all the way down to the bottom. Starting my timer there. I need to close this up a little bit for you guys, but my first one is in. Trial number 2. I'm going to get my filter paper disc going. I'm going to drop it in. Wait for it to go down to the bottom. Start my time. Now I have trial number 3. So this is all for pH of 7. And you can see our first trial is actually making its way up already. And we're going to say time. So 43 seconds. Trial number 3. We're going to stick trial number 3 in right now. We'll come back and talk about trial 1. Put that in. Let it drop down to the bottom. Click my time. So trial number 1 was already done. It was 43 seconds was my total. We're waiting for trials 2 and 3. So a little bit quicker than what we saw with pH of 5, and definitely quicker what we saw with pH of 3. So the enzyme definitely likes this particular pH just a little bit better, but we still have pH of 9 as well as a pH of 11. Here comes trial number 2, making its way up to the top, kind of stuck in the middle. We'll wait for it. It needs a few more oxygen bubbles to get trapped on it. It needs more of that reaction to occur. So it's kind of stuck in the middle right now. We don't want to disrupt these tubes or move them around any. Here it comes. Keeps going, keeps going, keeps going. And here comes the third one as well. Third one's bolting up. Now it's floating down. Second one's done. 1 minute, 30 seconds. We're just waiting for that third one now. Third one started making its way up, and then it kind of stopped. And it's making its way up now. And we're going to say time. 1:22. So there are my 3 times for a pH of 7. I have 43 seconds was my first one. My second trial was 1 minute, 30 seconds. My third trial was 1 minute, 22 seconds. So that was a pH of 7. I have 2 more pHs that I need to test, and then we will look at my 4 temperatures. Put this over here, get a clean shot here. We're going to reset all of my timers for trial 1, 2, and 3. Reset the second one, reset the third one. So as I go through, I'm now going to go to a pH of 9, So slightly basic now, just a little bit up that pH scale on the basic side. So we have more of those hydroxide, those OH ions than we do hydrogen ions. There's trial number 3 for a pH of 9. Here's trial 2 for a pH of 9. And my trial 1, and we're still doing everything else the same. I'm still going to do my yeast solution, my living organism that has the enzyme catalase in it. And we want that catalase within my yeast to break down the hydrogen peroxide so that it can produce the little oxygen bubbles and bring it up to the top. We want to see how well that yeast solution is physically working. So pH of 9, I'm sticking my first one in. I'll let it go to the bottom. Once it hits that bottom, I'm starting my time. I'm going to go to my second one. Make sure you only have one disc. It'll change the mass. It'll change how much yeast is going in there. Second one's going to go in. I'll start my time for the second one. Now I need trial number 3. Once again, this is pH of 9. And I'll stick that third one in there. Let it go to the bottom. Once it hits the bottom, that's when I'll start my timing. So there we go. We'll let those 3 run, and we want to see, are they going to be faster or slower than my pH of 7? And then we got one more pH once we are done this. And that will be my pH of 11. So all of my yeast discs are on the bottom, hopefully working upon that hydrogen peroxide, which is the substrate, the catalase, which is in my yeast. My yeast is producing it because the yeast is a living organism. That's the enzyme that's going to work upon the substrate. And the products that it's trying to form is oxygen, which is going to trap to my little discs. And you see the one there in the middle that's trying to jump around. The one on the end is trying to jump around. And that oxygen that gets trapped on the disc is going to bring the disc back up to the top. And that's how we can see how quickly this reaction is working. We want to see what are the effects of my pHs on them. So disc number 3 is actually making its way, kind of stuck in the middle right now. You guys can see that right there. So he's kind of stuck in the middle. My other 2 are just kind of hanging out as well, not really going too far. Disc number 2 is making its way up to the top. Disc number 3, disc 2 is there. Disc 3 is there. Here comes disc 1. Okay, so what we have, if we're looking at trial number 3 for pH of 9, my time there, 118 was my first one. My second trial was 134. My third trial, I'm sorry, my first trial, because we put that one in first, was 2 minutes and 11. So trial number 1 for pH of 9, 211. Trial 2 for pH of 9, 134. Trial 3, 118. So I'm going to write those down on my data chart real quick. We are going to be averaging them, and you guys will be graphing the average. And my last one, like I said, was 118. So not as quick as my pH of 7, but it still had a pretty good reaction time. And now we want to check a pH of 11. So now we're going super basic on that far end there. And I want to fill it up with my pH of 11. So it's got some hydrogen peroxide in it, and it's got some sodium hydroxide to make that particular pH. So if we're looking at that pH scale of 0 to 14, we are right now on a pH of 11. And how does this pH affect my enzyme action? Does it slow it down? Does it speed it up? Does it stop it altogether? So now we are on my pH of 11. I'm resetting all my timers here for all 3 trials. And I'm going to go back to my yeast solutions, my little filter paper discs. Dip them in. Excess off. I'm going to drop the first one into my pH of 11. This is trial number 1. When it hits the bottom, I hit my timer. We are starting. Max time here is going to be 4 minutes. My second one. And now we're doing all 3 trials. Start the timer when it hits the bottom. third one. Same thing, pH of 11. We're just getting our averages because there are many factors that may change here. So the amount of yeast that's on those particular filter papers, so I had 2 stuck together, things like that can affect it. So I'm letting that time go, giving my little yeast guys some more warm water so they can be happy. They tend to thrive better in warmth. So those pHs of 11, those discs are still at the bottom. Putting my yeast container back into some more warm water so that those yeasts can start to grow just a little bit. And we are at 1.23, 1 minute, 23 seconds, a little over a minute, just about at a minute for trials 1, 2, and 3. We want to see how they work in pH of 11. So 4-minute maximum time. Once this particular trial is done, you guys should now have a data chart for all 5 pHs. We did pH of 3, 5, 7, 9, and this is 11 right here. You guys will be averaging each one of your pHs, and it's the averages that you are going to graph. It tells you about the graph on there. So when you guys are making that graphs at this point should be done on the computer. If you're not sure how to do it, contact me. We can go through it. If you need a little Google lesson on it, or if you need a YouTube video that might teach you how to do that, please do so. But it's what you're graphing are going to be the averages for each one of these pHs. So our pHs are what you are controlling. Your reaction time you cannot control, so that will vary. So think about what needs to go on the independent and dependent axis here. So we're about 2:45 on the first one, 2:25 on the second one, and a little over 2 minutes on the third one. So pH of 11 is very acidic. We're not going too far with these, but we're going to give them a 4-minute time frame just to make sure. And think about what's happening there with the enzyme substrate complex. So when the enzyme catalase comes up with the substrate, my hydrogen peroxide, they should be forming a product. There is no product being formed here right now, and I know that because my discs are not making their way to the top. If the enzyme and substrate were working together and producing a product, I would have water and I would have oxygen bubbles. And those oxygen bubbles would be attaching to my filter paper disc, making that disc rise to the top. But at this point, those discs are not rising. We're still going to give the first one another 30 seconds. This one's got about another 45. Last one's got about another minute before we cut it there. So we said 4-minute maximum was our time. So we're about 3:45 on the first one. Really haven't seen too much movement whatsoever on it. No bobbing up and down, no kind of floating halfway. I don't really see too many oxygen bubbles being produced. There is hydrogen peroxide in this solution, but what's going on with the pH of 11 that's affecting this reaction? So my first one, we're going to stop it. We're already past 4, so we're going to say 4 minutes for the first one. We're just about 4 minutes for my second. That disc is still at the bottom. We're going to end that one. We're going to say 4 minutes. Clear that. We'll clear my first one. And we are at 4 minutes for the last one. So my discs are still at the bottom. My catalase was not able to work. What about that particular pH that did not allow it to work? Okay, so we're going to stop the video on this part here. We're going to come back in a few moments and we'll talk about our temperature portion of it.